In this experiment, we will be analyzing the biological principle of photosynthesis. Photosynthesis is the process of converting light to chemical energy. For this reaction to occur, the plant requires water, carbon dioxide, and chlorophyll. With these, the process results in the products of glucose and oxygen. Photosynthesis occurs in plants and some algae. The process of photosynthesis takes place in the chloroplast, specifically using chlorophyll, which is the green pigment in leaves. Photosynthesis applies to animals, for it is essential for their survival. It produces the sugar and carbon that enter the food chain, as well as the oxygen used in respiration. Plants are completely reliant on photosynthesis as their source of energy production, and without it they are unable to sustain life. Photosynthesis is a cellular reaction, but this principle has no role in every cell, like the principle of diffusion does. The process of photosynthesis is much like the process of making a protein shake. First of all, you need your ingredients, milk, which is much like your H2O. Second of all, you need your carbon dioxide, an egg. The third ingredient is a carbon, or chlorophyll, which facilitates the reaction. The fourth ingredient is light. Light is the exogenous energy that is required for photosynthesis to occur, light being the shaking of the protein shake. The end result of photosynthesis is starch, which then provides energy for the food chain. The aim of this experiment is to test whether carbon dioxide and water are vital for photosynthesis, which is hypothesized that they are both essential. 3 of the same plant, crepe myrtle, were used in this experiment. The plant was initially left in a dark space for 48 hours to destarch the leaves. For the plant void of carbon dioxide, it was initially filled with water. Potassium hydroxide was then placed in this jug. Glad white was wrapped around this plant and secured tightly to the top of the jug. Control received the same amount of water. All plants were then exposed to the same amount of sunlight for 1 week. The plants were then tested for starch. Its presence in the leaf would highlight that photosynthesis had occurred under its condition. To test for starch, the leaf was firstly placed in boiling water to soften the leaf and break down the cell wall. This boiled water then acted as a water bath for methylated spirits. The soft leaf was then placed in methylated spirits so as to remove the chlorophyll from the leaf. Once the chlorophyll was completely removed, the leaf was replaced back in boiling water so as to soften it for the absorption of starch. Once resoftened, the leaf was removed, placed on a clear surface, and flattened. 1 dropper of iodine was placed on the leaf to show up starch. The leaf was then compared to a darkness rating chart, with the higher the number being the more starch present. This was repeated for all 3 leaf conditions 3 times. The control contained the most amount of starch with a rating of 4. The plant void of water contained almost no starch with an average rating of 1.3. The plant void of carbon dioxide contained no starch with an average rating of 1. Results saw a consistent lack of starch in the plants without water and without carbon dioxide. These results prove the hypothesis to be true that you need both carbon dioxide and water to perform photosynthesis.